Hi, good morning and or good afternoon, everyone. Thank you for joining us. My name is Boren. I work for Opentrons as a scientist. Today's topic is about ELISA and how we automate this assay using a liquid handler. So we'll first introduce briefly how ELISA works and then show you the workflow when we run in the ELISA on a liquid handler. We will go through the assay results that support the idea of automating these experiments. So ELISA, or sometimes called EIA, is considered the gold standard of ELISA assays, of immunoassays. This is a very sensitive test to detect and quantify a target molecule. Some example of ELISA testing include HIV diagnosis, pregnancy test, and blood typing. It is also a popular method used in research labs when we need to identify the presence of a target of our interest in the samples, and if there is an antibody available for that target. There are several formats of ELISA, but in general, ELISA is developed around two biochemical reactions. First, there must be a antibody-antigen interaction, so the target can be selectively separated from other non-target molecule in the sample. And then we need an enzyme that can produce a signal, which can be a color, luminescence, or fluorescence. The drawing showing here outlines the steps to run a typical sandwich ELISA. An antibody specific to the antigen is attached onto the solid surface. In this case, the antibody is coated on the bottom of a well of a 96-well plate. The sample is added to this well, followed by incubation at a set temperature to allow the antibody to bind to the antigen. And then the well will be washed for a few times to remove whatever in the samples that are not captured by this antibody. And then the second antibody, also specific for this antigen and labeled with an enzyme, for example, a horseradish peroxidase, is added. Again, there's an incubation at set temperature followed by several runs of washing. Now the antibody-antigen sandwich is formed, and we can start a chemical reaction by adding the enzyme substrate into the mixture to generate the color that can be measured by a plate reader. So again, there's several formats of ELISA developed for different applications. But in general, the major steps in the procedure are liquid transferring of samples, reagents, and wash buffer, incubation, and mixing or shaking. So we will present several ELISAs with different formats or different targets, but the method for signal development was the same. It was carried out by using horseradish peroxidase as the enzyme and TMB as the substrate. It generates a blue color and then turn into yellow after stop solution is added. The absorbance is measured at 450 nanometer of wavelength. So those major steps can be performed on a liquid handler, such as liquid transfer, washing with a multi-channel pipette. And the incubation can be carried out on a temperature control shaker integrated on the platform to streamline the process. The liquid handler can also be programmed to do sample or reagent preparation before ELISA. For example, to make serial dilutions of sample or to distribute antibody solutions or wash buffers from the stock to 96-well plates, which can be stored for future use. In many cases, the ELISA kit does not come with a pre-coated plate, or we have to develop the assays in-house. In those cases, we can have the liquid handler to run plate coating as well. So the final step is to manually move the plate to the reader for absorbance measurement, or if we have a plate reader integrated, these steps can also be automated. The first ELISA we tested was Takara's fibronectin EIA kit. It was a sandwich ELISA using two fibronectin antibodies. One is coated on the A well strips provided in the kit. These A well strips can be assembled on a frame to a 96-volt plate. The second antibody, labeled with horseradish peroxidase, is prepared in solution. To run the ELISA, the first antibody captured fibronectin in the sample, followed by washing to remove non-targets, and the second antibody binds to the immobilized fibronectin, and the color is then developed and read by the plate reader at 450 nanometer. So this slide summarizes the workflow and the settings of the experiment. The protocol used a-channel pipette with 300 microliter tips. The heater shaker modules was integrated at the bottom right corner, and the antibody pre-coated ELISA plate sit on top of this the heater shaker. The reagents, including enzyme conjugate antibody solution, substrate solution, and stop solution, were in a 96-volt deep well plate, and wash buffer is in another 96-volt deep well plate. The samples were prepared by making serial dilutions of purified fibronectin in a 96-well plate at bottom left corner. And the system was programmed to follow this workflow, shows on the left-hand side of the slide. So the first step is to transfer samples to the ELISA plate, and then incubate for an hour for 37 degrees. So the pre-coated antibody in each well can bind to fibronectin, and then wash the plate for 3 times, meaning transfer wash buffer into the plate and then remove it for 3 times. And transfer enzyme conjugate antibody solution to ELISA plates. Again, incubate for another hour for 37 degrees, so the sandwich complex can be formed. And wash the plate for 3 times. Add substrate solutions, incubate 15 minutes at room temperature, and then add stop solutions before taking the plate to a plate reader. So the plate is sealed to prevent the evaporation during incubation, especially when the higher temperatures, such as 37 degrees, need to be maintained. And this slit seal allowed the pipette tips to into the well to do liquid transfer, and the latches on the side of the heater shaker close to secure the plate. And the washing step is critical for reducing the background signal due to the unbound antibody. We position the pipette tip to the side of the well when removing the liquid. This way, the residue liquid, and also called a depth value, can be significantly reduced to improve the effectiveness of washing. We did another ELISA, which is a competitive ELISA from Tecan. And in a competitive ELISA, a reference antigen is added to compete with the target antigen for the binding to the antibody. This reference antigen carries the enzyme responsible for the signal development. The higher the target antigen is present in the sample, in this case, cortisol, the fewer the binding site can be available for the reference antigen to be captured. So less reference antigens stays in the well after washes. Therefore, less signal is produced. In other words, the absorbance is inversely proportional to the cortisol level in the sample. Since this assay did not require a second antibody, the process was simpler, but it still followed the general workflow such as reagent transfer, incubation, washes, and then signal development. According to the assay instruction, the incubation did not need to be at 37 degrees, but require continuous agitation. So again, the ELISA plate is sealed and secured on the heater shaker. So this is another test. This test kit is to measure COVID neutralizing antibody, and this is obtained from Cell Sciences. It is a different format of comparative ELISA, which the antigen instead of antibody is coded on the plate. The reference molecule ACE2 carrying the enzyme compete with the target. ACE2 is a receptor present on the human cells, and the target here is the neutralizing antibody that binds to RBD. RBD is the receptor binding domain of viral spike protein. The workflow was similar, but the incubation was carried out at 37 degrees without shaking. So as you can see, depends on the assays or depends on the vendors who are producing these assays. Sometimes the incubation need to be at 77 degrees, and sometimes it need to be shake. So our system can be programmed accordingly. So we have introduced 3 ELISA kits tested for automation, which were designed based on 96 well settings. It is becoming more popular to use magnetic beads as a tool for separation. In this case, the beads serve as the solid surface for the antibody or the antigen to attach. The advantage include an increased surface area and an even distribution of beads throughout the sample processing, which can improve sensitivity to help detecting the target when the target is at low concentration. We have also developed protocol to run bead-based ELISA. And for example, this one is a RBD conjugate magnetic beads for COVID neutralizing antibody test. And the reagent is from Acro Biosystems. The beads are resuspended in the sample, allowing the RBD to bind to the neutralizing antibody. And since this neutralizing antibody are human IgG, an antibody against human IgG carrying the enzyme is then used for signal development. And during the washing steps, the wash buffer is added and mixed, and then the whole complex can be moved aside with the presence of the magnet, so the supernatant can be removed. And in addition to the pre-coated beads that we purchased from the vendor, we also made some RBD conjugate beads in-house and tested the protocol for automation. We include a magnetic module on the deck for bead-based separation. The workflow is still pretty similar to a typical plate-based sandwich ELISA. The sample plates was manually moved the heater shaker when incubation was required. But if your liquid handler has a robotic arms to move the plates between deck slots, this step can be automated. And all the assays we presented today were performed on the Opentrons OT-2 system. A P300 8-channel pipette did all the liquid transferring. The heater shaker provided temperature control and mixing by agitation. And magnetic module here was used for bead-based ELISA. So the next few slides will quickly go through the data that confirm the quality of the sample processing on this liquid handler. We first ran the EIA kit for fibronectin detection. The samples were prepared by making a serial two-fold dilution of purified human fibronectin. On the left-hand side, the chart shows almost perfect positive linear correlation between the assay's result and the target molecule concentration. Each data point was the average of triplicate result. They all have a coefficient of variation or CV values lower than 10%. The experiments was repeated for 2 more times, showing a reproducible result as these 3 response curves were almost overlapping, shown in this right-hand side figure. And this slide summarized the result from ELISA for cortisol test. This is a competitive ELISA, so the signal produced is inversely proportional to the cortisol concentration in the sample, which again prepared by making two-fold dilutions of purified human cortisol. The number next to each data point represent the CV value, and all these values are lower than 10%. The chart was plotted for the readout versus cortisol concentration at log scale, showing a negative linear correlation. All 3 experiments generate consistent results as shown on this chart on the right-hand side. These are the results from a COVID neutralizing antibody ELISA. It's a plate-based ELISA. We purchased human serum samples which have been determined antibody positive or negative. And since this was a competitive ELISA, a lower absorbance indicate a higher level of COVID neutralizing antibody. The quality of the assay processing was confirmed as these CV values were lower than 10%. The signal produced was further converted to inhibition rate according to the formula provided by the vendor. So the higher the inhibition rate is, the more neutralizing antibody is present in the sample. The inhibition rate higher than 20% are considered neutralizing antibody positive, which agree with the results previously tested by FDA certified ELISA. And we did 3 different 96-well plate-based ELISA, and then we move on to bead-based ELISA. To create standard curve for the assay, we prepare serial two-fold dilutions of anti-RBD antibody, which is the target. The CV values look great, and the positive linear correlation with R square of 0.999 was shown by plotting the target concentration versus readout. And we ran the protocol again to process RBD attached dyna beads we prepare in-house. And as we expected, the CV values confirm the consistency of sample processing, and the standard curve look very sharp. We then ran the protocol to process the serum samples to measure COVID neutralizing antibody. The assay was able to confirm positive samples and quantify the level of the target. The results have again demonstrated consistency of sample processing on the liquid handler. So this slide summarizes the settings and the runtimes of the assays we presented today. All the protocols for automation were developed by converting the assay instruction included in this ELISA kit, which are usually performed by hands. Some assays require shaking during incubation, and some assays require incubation at an hour at 37 degrees, but some can be incubated at room temperature. So the runtime is largely depending on the design of the assay. For example, a typical sandwich ELISA having 2 1-hour incubation will take longer time to complete. And again, so with the heater shaker and magnetic module integrate on the platform, together with multi-channel pipette that provide high-quality sample handling, so all the ELISAs tested were successful with excellent precision and accuracy. Automation can be a labor-saving, a cost-effective solution for ELISA, especially when we have to process larger throughput numbers of samples. So that will conclude our presentation today, and thank you for your attendance.